Welcome to UC Science Radio, where we conduct interviews with a range of scientists to learn about the big issues facing our world and what science is doing to help. I'm Molly McGidd, a master's student in the School of Biological Sciences. Today, I'm talking to Dr. Tammy Steeves. She's an associate professor in biological sciences and works to preserve the genetic diversity of some of New Zealand's rarest taonga species. She does this research in collaboration with relevant Māori communities and with conservation recovery groups to put her science into practice. Kia ora, Tammy. Thanks for joining me on the UC Science Radio. So, what do you do at the university and what research do you do?
Kia ora, Molly. Thanks very much. I do a lot of things at the university, like most academics, but mainly the research that my research team and I do is focused on two main threads. So one is to use genomic and non-genomic data, so basically a lot of DNA material to address conservation management questions for critically endangered species. And then also the other aim of what we do is to use similar types of data to inform things like conservation translocation decisions for threatened freshwater species. And so generally there, what we're thinking about is how and where and when and why should we actually be moving species around.
And something that I've heard you describe yourself as is a genetic matchmaker. What does that mean?
A genetic matchmaker, or Tinder for birds, I think we've used as well. So one facet of the work that we do is it can be really challenging to pair together individuals in small conservation breeding programs, often because we don't actually know who the founding individuals were that started out that program. And so essentially what we do is use that genomic or DNA data to estimate relatedness among individual birds. And then we combine that with other data to basically make pairing recommendations. And we work directly with conservation practitioners to then go on and actually act on those recommendations.
So you're pairing up individuals that would best be suited to preserve genetic diversity? Is that the idea?
Yeah, definitely. Yep. So in conservation breeding programs, what we're seeking to do is to reduce inbreeding in the short term. So we don't want close relatives mating, because in the long term, what we're looking to do is to minimize the loss of genetic diversity. And the reason why we care about that is because species actually need diversity in order to respond to changing environments down the track.
So you mentioned that you work with some conservation groups. What is that like, translating your research into something that could be applied out in the field?
Oh, it's really rewarding. So I think in our research team, we have a number of mantras, but effectively all of us get a lot of joy out of being of some use. And so we do a lot of, all bias included, some really exciting research at the edge of what conservation management science looks like. But fundamentally, our work isn't really done until it's actually been translated into conservation management practice. And the way that we achieve that is in partnerships. So we work directly with conservation practitioners from the very, very beginning and throughout that process to come up with decisions that matter. And I've certainly been times when I've been known to say things like, you're not doing conservation genetics unless you're actually working directly with practitioners. And so having said that, the theory of conservation genetics is incredibly important, but the work that we do is very much on the implied end of things.
I also know that you co-develop a lot of your management practices with relevant Māori iwi and hapu. How does that work, and how does that process happen?
Yeah, it's a really good question. Fundamentally, like all things, it's about building trusted relationships. And depending on the species that you're working with, it can look a little bit different. So for example, here at University of Canterbury, we are on Ngaitahu Takiwa, or region, and in particular, the iwi or hapu Ngaitu Aotearoa. So for work that we do on species whose distribution includes Ngaitu Aotearoa's Takiwa, we work directly with them. And so, for example, we have a couple of projects, one on a nationally critical species called Kawato, or Canterbury mudfish, and then also a declining species called KKY, or freshwater crayfish. And so those are where we have those relationships directly with hapu or iwi, or effectively mana whenua. Having said that, for some species that are actually protected under the Conservation Act, if there are species recovery groups associated with them, for example, within Ngāi Tahu's Takiwara region, there are Ngāi Tahu representatives on those species recovery groups. And generally those are for quite high profile species. We work on kaki or black stilt and kakariki karaka or orange-fronted parakeet, for example. And so we work directly with those representatives as we travel in the research that we do.
Could you talk a bit more, maybe give an example of species that you work on and what sorts of strategies you've co-developed?
I think, yeah, it's a very good question. So for example, with one particular project, so the work that we do again is we often generate genomic data, and that might be generating what we'll call a reference genome. And so we often say when we're looking at genomic data, we're looking at sort of pieces of a puzzle where the complete puzzle represents the whole genome. And often when we're working with those small puzzle pieces, it's great if we have a completed puzzle to refer to. And so that's when we generate something called a reference genome. And so, for example, we've made decisions in partnership with Ngāi Aotearoa around where we sample individuals to generate reference genomes, how we sample them, where we generate the data, how we generate the data, where the data is stored, and generally package that around the narrative in terms of what are our research questions to start out with, and then also how will we then share the results of those research. So one example that we've used before is kawaro, again a nationally critical species, quite possibly one of New Zealand's most threatened freshwater fish, not protected by any sort of Conservation Act, for example. And that species, we wanted to sample from a site of local significance, so from a nearby area called Tuhaitara Coastal Park. But the work that we did there showed that there were actually not very many kawaro in the region. So the last thing we wanted to do was to sample an individual from that site, despite its cultural significance, given the low number of samples. So what we then did is we went back to the kaitiaki from Ngāi Aotearoa that we work with, and we chose a different location with a much healthier population from which to sample an individual to generate the reference genome.
Yeah, that's really interesting, being able to, balance different important reasons for doing certain things and making sure that, you're doing the science responsibly, but then also thinking about all these other factors.
Yeah, absolutely. They go hand in hand.
Great. So what is the most fun or exciting thing about your work? Any part of the work?
Well, I absolutely love working with the team of early career researchers that I work with, and I absolutely love working with our partners, whether they're hapu or iwi, or their Department of Conservation staff, or others who work in that space. I'm an inherently collaborative person, and I really, again, lots of mottos in the group, but it takes teamwork to make the dream work. And the sum of all of our parts is always greater than what we could achieve on our own. So we have a lot of fun in the group, but the standards are also very high. And that's fantastic, because when you're doing excellent work with a group of excellent people and you're generating science that has genuine impact, it's fantastic. I feel really privileged.
And on that note, I know that you are involved in, is it an initiative or a group called Kindness in Science that sort of pushes for this idea of community and kindness to achieve better outcomes in science. So could you talk a bit about that, how it was created and what its mission is?
Sure, yeah. So Kindness in Science started with a conversation, like they always do. It was myself and my former PhD student, Stephanie Gala, and a former postdoc here at University of Canterbury named Catherine Fabria. And generally what we were talking about, with me as a mid-career academic and a PhD student and a postdoc student who are both early career researchers, was sort of just reflecting on a lot of the unkindness in the science system, and generally asking, there must be a better way. Is there a better way? And what spurred that initial conversation was actually a paper, a short editorial in Nature, where a woman spoke about the microaggressions that she experienced in her workplace. And the response to that was just downright horrifying, with people minimizing her experience and generally just being downright nasty and bringing with them a lot of misconceptions about what it means to be a scientist and how we can create and do better science. And so what we reasoned quite quickly is that we'd sort of settled in on a perfect storm. There were a number of people around the world, but also here in New Zealand, thinking about ways to make the science system more kind. And I really focus on the system in itself, or the institutional structure, because it's not really about just being nice to each other. That's absolutely not what kindness is, or being kind is. It's actually very hard to be kind, because it's about pushing for what is right and just. And I often talk about it in terms of making space at the science table, and that what we're looking to do is to make science an inclusive and diverse and accessible and equitable place. And the personal story for me goes with that my now 8-year-old daughter had made me a science table with my husband. And it was a nice flat lab bench looking science table. And then as we progressed in kindness and science, I'd said, we just need to smash the table altogether and build a brand new table that has dips and bumps and reflects the diversity that we want to see at this particular table. But of course, when your 8-year-old makes you a table, you're not going to physically smash it. So what she and my husband then did was built me a new table. So in my office, I now have 2 science tables, one which reminds me of where we started, and the other which reminds me where I hope we're headed.
And so it seems like these efforts maybe started out as being focused more on postgrads and later career scientists, but how do you bring inclusion early on into the science classroom at universities, or even with younger students, to make sure that people feel like they belong in science? Do you have any insight on that?
That's a very big question.
It is.
But it's a very good one, because if I reflect a little bit about what I've learned, I'm no expert on this at all, but it's certainly some of the things that I've learned and heard as we've moved forward in this space is, so for example, within a New Zealand context, you may hear academics lament, oh, I've got this PhD scholarship or this postdoc that's available for Māori students. Where are all the Māori students? Why aren't they here? Or even as undergraduates. And where might those students be? And some of the things that I've learned along the way is that if we actually want to target representation by the time we hit the tertiary sector, we really need to be targeting communities much, much earlier as primary, in terms of introducing science and what it is, and the fact that a scientist doesn't look like any one thing, and breaking down those stereotypes and providing clear and sustained and meaningful pathways into science. And that includes supporting communities to see themselves in science as well. And so you'll often see targeted programs that might be targeted at year 10 students or targeted at younger students. And I think all of these initiatives, I see them as being yes and initiatives. There's no magic bullet to creating a safe and inclusive and diverse space. But there's one way not to do it, is to just stand around and wonder where the hell everybody is, because there's a damn good reason why they're not here, or a whole series of reasons that start really on when we think about inequity in society. And so it's a really big ask. But what I think we look to do is not get overwhelmed by all of those things, but think about, I often talk about using your privilege for good. And we all have spheres of influence. And so for all members of the Kindness and Science Collective, whether we're PhD students or master's students, or high-level managers at universities or within human resources, or professors, I encourage people to contemplate their spheres of influence and use their privilege for good within that sphere. Because even spending 10 minutes with that one person in that one interaction could be life-changing, Or it could be that in your sphere of influence, actually, you need to apply sustained pressure to the people who create and sustain change in an institutional setting, for example. That's what's necessary to actually change the science system. So yeah, that's a very big question, and I hope I've just touched a little bit on it.
Yeah, absolutely. Yeah, it is a big question, and it's going to take time because I think as many people have encountered, the science system, unfortunately, often tends to be competitive when funding is limited and people are applying for it. There's sort of a drive to be more separate and individualistic about how you go about things. And hopefully that's changing. And I think I've experienced that it is, but it's going to take a while to sort of break down those preconceived notions. And they go all the way through different sectors of science education. So it seems like a really valuable collective.
Thank you. Yeah, we've got lots of work to do, for sure.
For sure. So my last question is, if you could say in a sentence or a couple of sentences why the work that you do and your group does is so important.
Oh my goodness. You're asking me for my elevator pitch, I think. Generally, I think what I would say is that the work that we do is we're starting the way that we intend to finish. We seek to do research excellence in a space that, particularly in a New Zealand context, where we honor the Treaty of Waitangi, and we are at all times seek to be excellent treaty partners, and to do that in a space where we're actually doing research that has impact. And we're not talking about impact factor, we're talking about research that actually impacts decision making.
That was excellent. Thanks for talking to me, Tammy.
It was my pleasure. Thank you.
That's it for today. For more information or to ask a question about any of the ideas raised in this show, feel free to send UC Science a message by email, Facebook, or Instagram. Thanks for listening.